everybody, so this is my practicum presentation. The title of my practicum is Using Dispersin B mutants to Disperse Staph epidermidis biofilms. My name is Kathleen, I'm a super bad kick. A little bit about me is I'm a sophomore and I'm in Science and Global Change, and I'm a biochemistry and microbiology double major. For my practicum, I volunteered at a biochemistry lab. The particular project that I'm going to talk about took place between the summer and fall of 2021. As an aside, I still work in this lab and I still work with this protein, so my practicum did lead to kind of like a longer term connection. For my practicum, I worked at the Colon Lab at UMD. My site supervisor was Dr. Alex Peterson, who recently defended, which was really, really cool to see. We investigated how changing some amino acids in dispersin B, which is an enzyme important in the dispersal phase of the biofilm life cycle, how changing amino acids in that enzyme changed how it could break down biofilms. The way we did that was we used biofilm assays. This is an example of a biofilm assay. This is a 96-well plate. In each well, we grew a biofilm and put either a mutant enzyme or different concentrations of an enzyme to see how it could break down the biofilms. The more purple in the wells, the more cells remain, the lower the enzyme dispersed the biofilm. So for my activities that I did during my practicum, I did a lot of typical biochemistry lab stuff, like dealing with cell cultures, purifying proteins, running DNA and protein gels, and obviously I did a lot of these biofilm assays. My particular learning outcomes was I obviously learned a lot about dispersin B, the protein. I learned that science is a process and takes a lot longer than I thought it did. I also learned personally that I like doing research and that I like working with bacterial cells and cell cultures. If you want to learn more, I would recommend reading over my practicum poster, checking out the Colon Lab website, or taking a microbiology class if you're really interested in bacteria and bacterial biofilms. Thank you so much for your time, and thank you so much for listening.